Now let us consider what is there in the complex number 5, that is ATP synthase, that is chemi-osmosis has to take place. And I use the word osmosis, that means something must be related across the gradient, there would be a movement, and that would be accompanied with the complex number 5, which is present coupled along with all these complexes wherever there is movement of proton outside the membrane. Now whenever the movement of protons is taking place outside the membrane, you saw it in the earlier 4 complexes that the protons were moving out in order to enable the electron transport. Those protons, when they move out, there is a gradient created. That means the protons which have gone outside, they would try to move inside according to the osmotic theories that say that whenever the concentration is high at one side, they have to go wherever the concentration is less, or you can consider it in terms of free energy. So the protons have to come inside the matrix, and they come inside the matrix through F0 and F1 particles, which is ATP synthase. When the protons are moving across their gradient through this F0 F1 particle, this ADP along with inorganic phosphate is converted to ATP. So in a way, the proton movement across the complex number 5 enables the generation of ATP, and for each time, every time whenever there is movement of 3 protons across the ATP synthase, there is formation of one ATP molecule. Single ATP molecule would be formed whenever 3 protons would move across the F0 F1 particle, and there would be generation of one ATP molecule which is being generated now by NADH and FADH2 only, because they are the ones who have started the movement of protons across the membrane. Now when this gradient, in order to break this gradient thing, when the protons come across this, 3 protons would move and one ATP molecule would be generated. So we get in a way that 1 NADH, sorry, I beg your pardon for this, 1 NADH would give rise to 3 molecules of ATP. That means in a way 1 NADH would enable 6 proton transfers. Not 6, it would be 3 proton gradient movements across the ATP synthetase would be enabled by NADH2. In the same way, 1 FADH2 would give rise to 2 ATPs. So you see there is a little bit of complexity in understanding that how the hydrogens were moving, and if we try to equate the thing, that is a bit complex, but all you have to understand is that this is coupled along with the movement of electrons. This complex is coupled with these movements, and wherever there is proton gradient and whenever that gradient is being broken down, ATP is being generated. In loss of a proton, 2 protons by NADH, single loss of proton, it would enable 3 times the coupled reaction where the ADP would get converted to ATP. So every time an NADH loses its proton, it is going to give rise to 3 ATPs. FADH loses its protons, it is going to give rise to 2 ATPs. So we reached to that equation where we had to reach 36 ATP generation. This is how the entire process of aerobic respiration is done. We did get to know that what is phosphorylation. That was the ultimate aim, was energy generation. Why do we take food? We take food so that we can get energy. That was by breakdown of this glucose, whichever was consumed by any organism or any cell, to be specific, whether it is a plant cell or an animal cell. It would take the food in this form, it would get broken down into these two things, and it would give energy. So the first question, that was what is phosphorylation? The production of ATPs in the presence of oxygen. The oxygen is required as the ultimate acceptor of the electrons which have passed the electron transport system. And the last one is, what is gradient driven ATP synthesis? It is the electron transport system which enable the generation of ATP with the help of complex number 5, which works on the chemiosmosis principle, that is breaking down of the proton gradient would give rise to ATPs. So we did see that how the NADHs were NAD positive got reduced to NADH. Later on, the reduction that took place by the addition of these protons in NADH2, that also took some sort of energy. That energy is to be given in the form of ATP when the protons that it took up during the Krebs cycle and the other process of glycolysis, it would give it back in the form of ATPs by losing these protons. These protons would come and break the gradient, and in this way we will have the required ATPs whichever are to be needed. Now this process entirely is about the oxidative phosphorylation, aerobic respiration we are talking about. But at times what happens is that the glycolysis is not followed by Krebs cycle. Krebs cycle is not followed by oxidative phosphorylation. So that condition is the process of anaerobic respiration that we need to understand. Thank you.